What does it take to be a successful athlete? I bet you're probably thinking of hitting the gym and building your muscle mass and strength, right? Most people think that the perfect recipe is a mix of intense training, balanced diets, and discipline. And while this is true, what if I told you there might be a key ingredient in your DNA? Say hello to MSTN, a gene that might be your golden ticket to sport success. Welcome to the genetic basis of stuff and things. After intense training, your muscle fibers experience tiny tears, which triggers a repair process. Muscle stem cells, known as satellite cells, are activated and transformed into myoblasts. These myoblasts then fuse with the damaged fibers in a process called hypertrophy, leading to bigger and stronger muscles. But this process requires regulation, and that's where myostatin comes in. What exactly is myostatin? Well, it's a protein encoded by the MSTN gene, known for inhibiting muscle growth and differentiation. Also called growth differentiation factor 8, myostatin is almost exclusively expressed in skeletal muscle cells. How does myostatin control muscle mass? Myostatin begins as an inactive protein. Once in the bloodstream, it's broken down into its active form. This active myostatin binds to receptors on muscle cells, triggering a signaling pathway that leads to protein breakdown and represses muscle growth. Essentially, myostatin ensures that muscles don't grow too fast or too large. But wait, how could a negative regulator like myostatin be important in sports? In 1997, researchers knocked out the myostatin gene in mice, and the results were astonishing. These mice exhibited double the muscle mass throughout their bodies. But does this also happen in humans? Yes. In 2004, researchers discovered a German toddler with unusually large muscles. And guess what? He had a homozygous loss-of-function mutation in the myostatin gene, meaning both copies of the gene were mutated, preventing myostatin from functioning properly. Interestingly, several of his family members were also reported to be unusually strong. This discovery opened up questions. How common are myostatin mutations in elite athletes? And could these mutations influence their athletic performance? While loss-of-function mutations in myostatin are very rare in the general population, geneticists focus more on common genetic variations called polymorphisms to better understand athletic traits. One such example is the myostatin lysine 153 arginine polymorphism, where a lysine is substituted with an arginine at position 153 in the active myostatin region. This is the rare arginine variant. This change in the myostatin gene may have an impact on how the precursor protein is processed and how well the mature myostatin binds to its receptor. What's fascinating is that this missense substitution is also found in other mammals, suggesting that it could have an important impact on myostatin's function. Even so, this variant is still rare, appearing in only about 3 to 4% of Caucasians, and fewer than 1% are homozygous. Because of this, studying it in large groups is challenging, but some findings are promising. For instance, in African Americans, this variant has been linked to greater elbow strength. Interestingly, women seem to respond better to strength training, and Han Chinese men show increased muscle thickness, while in young Spanish men, it's even been linked to better power during jumping. However, not all studies agree. Some have found no significant impact on muscle growth, even in powerlifters. When comparing Israeli swimmers and runners, the variant was found to be more frequent in runners, especially among top-level sprinters. Surprisingly, although it was more common in long-distance swimmers than sprinters, it wasn't linked to top swimming performance. On the other hand, while the difference between athletes and non-athletes group wasn't statistically significant, the frequency of this variant in Israelis was still higher than in non-athletic populations from other countries. So, it seems that this rare variant might be particularly beneficial in strength and speed-based sports, and arginine carriers might have a genetic advantage for running success. Another interesting variation is the alanine 55-threonine polymorphism. This variation occurs in a key region of the precursor protein and has also been linked to sports performance. The location of this polymorphism suggests that changes in these regions might reduce the protein's ability to suppress muscle growth. This explains why those who carry these variants could benefit from greater muscle mass and potentially have an advantage in sports. Myostatin isn't the only gene related to sports performance. The first genetic polymorphisms linked to sports were in the angiotensin-converting enzyme and alpha-actinin 3 genes. Since then, hundreds of other genetic variations have been discovered. Athletic traits are complex, shaped by the interaction of many genes and environmental factors, like training and nutrition. Research in sports genetics remains controversial and mostly focuses on skeletal muscle traits. So far, genetic factors only account for a small fraction of what makes someone a successful athlete. However, with progress in bioinformatics and machine learning, we may soon develop tools to better understand the genetic basis of sport success. Indeed, what a great time to be a geneticist!